Thank you for the opportunity to present the results of a pilot study we recently conducted, characterizing associations between the gut microbiome and pain interference among 35 living donor kidney transplant recipients. As surgical techniques have improved and immunosuppression have improved, it's opened the door for a wider pool of patients who are more medically complex. And with that has come an interest in understanding better symptom burden after transplant and health-related quality of life. Chronic pain persists in a third to a half of kidney transplant recipients. Pain interferes with daily functioning, and it has potential serious consequences, including difficulty with medication taking, decreased enjoyment of life, increased risk of allograft rejection, graft loss, and mortality. Symbiotic microbes in the gut perform critical functions in health and disease, and new evidence sheds light on the role of the gut microbiome in mediating pain via the bidirectional signaling pathway between the enteric and central nervous systems known as the brain-gut microbiota axis. Briefly, we were pleased to learn that participants were from this diverse sample were willing to collect fecal samples. It was a big ask. As for the microbiome feature analysis, we first examined microbial diversity between those with pain interference and those without. These metrics give us an idea of how diverse the communities of microbes are in the gut. Greater community diversity is usually associated with better health. We did see a reduction in the Shannon Diversity Index in all groups. However, there was no statistically significant differences between groups. However, we did see some statistically significant shifts in microbial communities among the people who reported having high pain interference over time. Next, we examined the differential relative abundances of bacterial taxa. And what we saw was a five-fold reduction in the genus Akkermansia in the pain interference groups. This is important because Akkermansia is an important commensal bacteria that plays an important role in maintaining the integrity of the gut epithelial layer and preventing pro-inflammatory signaling molecules from entering the systemic circulation. We also observed an increased abundance in several pro-inflammatory taxa among the people in the pain interference group. This is important because the gut microbiome is modifiable and can be modified through changes in nutrition, physical activity, and stress reduction. We recently received a R01 from the NIDDK to further our study here, and we look forward to presenting that at a future meeting. Thank you.